Welcome everyone to Zakra's Biomedical Innovation web series. I'm Preeti Cletus and I'm the CEO of the Zakra Group. It is my pleasure to kickstart this session, which is jointly sponsored by Zakra Scientific and Predicin. We aim to bring to the forefront technologies that are boon to precision diagnosis and which complements the development and prescription of targeted therapies to patients. Just to give a brief introduction of Zakra, Zakra is headquartered in Singapore and is now present in several countries. It was sprouted with a clear intent to ensure that the latest high-efficacy biomedical solutions are made available for all. We recognize and are confident that these solutions are valuable towards supplementing quality healthcare and lifestyle. With that intent, we are purposeful in bringing to the community innovative and highly effective solutions that will fill or narrow the gap between the ailing and their means to a remedy. We in Zakra believe that in giving, we receive, and actualize this to three broad strokes: awareness building, action, and advocacy. Our ultimate goal is for every individual to be a believer of innovative solutions, so much so they become strong advocates to others, and in doing so help the community level up towards betterment of good quality life. For this to happen, we need to have key players like physicians, scientists, and leaders of our community to act in embracing technology and have these technologies serve the community. We need such actions to be far-reaching so that the tangible outcomes cannot be missed or sidelined, and in so being, garner an ever-increasing support and resources that is very much the lifeblood to serving the community. So this leads to our third stroke, which is awareness building. We can neither act nor advocate something which we are not aware of or know insufficient clarity about. Zakra's web series is our noble attempt to bring about this awareness and to allow practitioners and stakeholders share and banter about solutions that can value add to the society and the nation. So I hope that today's session will be insightful with a robust discussion that brings everyone to a notch higher towards a unified approach to serving the people. And now it is with great pleasure that I introduce our esteemed speaker for today, Dr. Manish Kohli. He is currently the Jack and Hazel Robertson Inaugural Presidential Endowed Chair in Cancer Research and a professor at the Division of Oncology in the School of Medicine of the University of Utah and Huntsman Cancer Institute. His accolades are far-reaching with a 24-year experience and still going as a medical oncologist with an entrepreneurial spirit and achievement. Now, before I hand over to Dr. Kohli, may I encourage participants to raise their questions via the chat function found at the top right-hand corner of your screen, which will be addressed during the Q&A session. So without further delay, I leave you in the expert hands of Dr. Manish Kohli. Dr. Manish, please.
Thank you, Prateep. I'm going to start sharing my slides. Let me know if you can see them.
Yes.
Okay. So let me just adjust my screen here and launch into today's presentation. Thank you for the opportunity to both the sponsors of this talk today. My title is in front of you: Molecular Profiling for Precision Cancer Therapies and the Role of quote-unquote NGS, which is an abbreviation for Next Generation Sequencing Liquid Biopsy in Detecting Actionable Biomarkers. I will proceed with my disclosure slide, which is to propose my conflicts of interest as listed here. And then I'm going to do today's presentation in three parts. I'm going to start with a very general overview of the concepts that are involved in tumor genomics. Then I'm going to progress to the task of molecular detection tools that we use in cancer. They'll very briefly touch on the pros and cons, and I welcome your questions in the Q&A session thereafter. Two broad categories of tools being PCR, which is an abbreviation, as you all well know, for polymerase chain reaction, and is typically useful for a handful of gene detections, versus next-gen sequencing, which typically involves far many more genes. And then finally, close it by giving a application examples of NGS-based precision oncology as we practice cancer treatments nowadays. Let's start with the first part. You're all very well aware of the genetic code of a normal cell, which has got 23 pairs of chromosomes in a double helix that is depicted over here. Each strand has billions, 3.2 billion base pairs, or these letters ACTG. Now, these letters of a normal cell multiplied by 2, because we are diploid human beings, undergo a normal process of cell division, which starts in the resting phase to G1 or growth phase, synthesis phase, SG2 or gap2 phase, and then mitosis. Mitosis is the place where number of things happen, wherein a normal cell divides into two daughter cells after a whole series of events that go through as listed over here. Now, this is the phase where lots of things are normal, but some things can go abnormal. In fact, when a normal cell divides into two daughter cells, at this particular point of the M or the mitosis phase, there are many things that can go wrong for the normal cell to become cancerous. In other words, instead of making two cells, it multiplies and makes many more cells. And what are those things that can go wrong over here? There are three broad categories of alterations, starting with mutations, wherein one of those ACTG letters gets substituted in a way that is horrible and becomes a mischief for the normal cell to transform into a cancer cell. Another kind of alteration is that instead of one gene, it may replicate into many more genes, which we call copy number gains of a gene, versus it could also go the other way, wherein the copy of a gene is deleted. We call that copy number deletion. And so copy number variations are aberrancies from the normal, and they could have deleterious effects on cancer. A third type of aberration, which you all very well know, which is the poster sign for precision oncology, is a gene rearrangement. During mitosis, there could be events like a distant part of a gene situated on a different chromosome, in this case BCR gene on chromosome 22, gets transposed into another chromosome 9, where another gene is sitting called ABL, and the BCR-ABL fusion causes a protein which is dominant to cause proliferation of the cell in a way that it leads to chronic myeloid leukemia, or CML. So broadly speaking, these are the alterations that can go wrong. And when these alterations go wrong, the transformation is at the level of the protein, where an aberrant or abnormal protein is formed. What is the implications of that? Well, the implications of that are that we are dealing suddenly with something called cancer. And cancer, therefore, is a product of these genetic alterations that take place. And let me illustrate that point in the sense of the genomic landscape of one particular tumor called non-small cell lung cancer. Previously, when we do a biopsy, we look at the morphology of the biopsy in a tumor mass and come up with this picture on the top left corner that you see. And we classify based on this picture. The pathologist classifies this patient to have one kind of lung cancer, either squamous cell or large cell or adenocarcinoma, which is the bulk of non-small cell lung cancers. But adenocarcinoma, which is the bulk, has got many underlying genetic reasons that it is formed into this picture. These are the list of some of the genes that we know already: KRAS, EGFR, EML, etc., which can cause this adenocarcinoma to look like this picture. Now, why is it important to know these genetic makeups? Let's take the example of one gene. The reason why it has become important to know whether EGFR, or epidermal growth factor receptor protein, is abnormal because of an alteration in the DNA of that particular gene is because we have targeted therapies now to these specific genetic alterations. Let me give you an example of that. EGFR mutations were first observed in 2004, and thereafter, in the next 3-4 years, it was enhanced to a drug called geftinib, which targeted that particular mutation in EGFR in non-small cell lung adenocarcinoma. Here is an illustration of the point. When you give a patient with an EGFR mutation a drug called geftinib, this tumor over here in the top picture disappears within a period of few weeks or months. And this cancer, this is the lung cancer in a CAT scan that you see on the top A and B pictures, while in the C and D pictures you see liver on the left side before giving geftinib, and after giving geftinib, this cancer has completely disappeared in picture D, and so on and so forth. And so it becomes important to know whether this adenocarcinoma has an EGFR mutation. Therefore, because we have a drug that can be targeted rather than give this patient very, very toxic chemotherapy. Another example of EGFR response in a lung cancer patient, on the top left is a lung on the right side, totally full of cancer. And then after giving the treatment, it completely disappears. And those are similar pictures again. So EGFR mutations in non-small cell lung cancer has been the source of a lot of discovery and invention since 2004. And now we have a bunch of mutations that we know which can be targeted by not one, but many, many EGFR based treatments called receptor tyrosine kinase inhibitors, of which geftinib is only one. Here is another mutation in another gene in lung cancer where a different set of drugs elicit a response where this particular tumor with this BRAF gene mutation completely disappears with a different set of drugs that are directed against this particular mutation. So you can get the drift of what I'm trying to say, that it is important now not just to throw chemotherapy on patients, but also to treat them intelligently based on not the morphology, but the genetic makeup of the tumor. And it turns out that as you travel from Western Hemisphere to the Eastern Hemisphere, EGFR mutations change in lung cancers in the amount and the frequency of those mutations. In the Western Hemisphere, EGFR mutations in lung adenocarcinoma range about 15 to 25% on an average. When you come to Europe, they are between that number and 17 and 30% or so. When you come to Middle Asia, it becomes a little more. When you come to South Asia, about 30 to 40%. When you go more east, it becomes about 40 to 50%. Now, if we did not know in these different regions how prevalent this mutation is, the advocacy of the drugs that have been invented would go wasted. And if we do not have the molecular tools to find those kinds of mutations, then we would not be able to decipher those mutations. And that's where all of this comes together as such. And not only is it the initial diagnosis and treatment for EGFR tyrosine kinase inhibitors directed against a specific EGFR mutation, but over time, what we have seen, there is evolution that takes place. You treat somebody with a particular drug smartly with a targeting a particular mutation. Over time, there is response, but then new cells which are mutating at a rapidly progressive way start appearing, which are resistant to this old drug because of a new mutation that comes. And now you have to target this new mutation with another new drug. And for all of this, you need molecular tools. And those tools are the ones that I will talk about next. But you get the idea of how we have changed from morphology to genetic basis and targeted therapies, and the diagnostic tests that are now becoming so important at every stage of the cancer treatment as such. And so in the past, we used to have no predictive biomarkers. Now we have predictive oncology biomarkers, which these molecular tools are developed on. We have markers which monitor for disease progression, markers which prophesize recurrence of the tumor or predict progression, and so on and so forth in oncology, which are very much part of the whole armamentarium and the ecosystem of treating cancer. In the past, the traditional way, where we used to take morphology, patient went to the physician, a biopsy was done, morphology was gotten, and we treated this patient with chemo or hormonal treatments, has come to now pass in the last 5-7 years, wherever these tools have been developed successfully, so that now the patient goes to the physician, a pathology, a biomarker and pathology biopsy is done, and not just morphology, but we look at the DNA of that particular morphology and then match a pharmaceutical drug against a particular mutation, and then we successfully treat our patients. So how do we get into the whole idea of diagnosing this case in the form of those molecular diagnostics? I'll speak to the second part of my talk of molecular detection tools, and I'll touch upon solid tumor biopsy versus quote unquote liquid biopsy in cancer very briefly. When we traditionally have gone forward and done biopsies, this is a case of a bone scan of a patient of prostate cancer who has a bone scan positive image that you can see over here as white. When we put in a needle that you see going into this bone, we end up getting a biopsy specimen, which is this big. This is measured in millimeters, and this is what we have to now look into molecular tools, which gives us very, very, very small amount of DNA, something in the matter of nanograms. In this nanogram is hidden the cancer biology of this particular tumor, and it is mixed with normal bone DNA also. And our job is to figure out with molecular tools that nanogram that is ascribable to the tumor as such. And for that, the tools that we have are things like polymerase chain reaction. Basically, when you have very small amounts of tumor, and this is a term that you have become very, very familiar with, especially in the last 2 years with the COVID pandemic, what it does, this chemical reaction, what it does is it takes the small amount, very rare amount of the target material that we are interested in, and through number of cycles, it propagates that one copy into hundreds and thousands and millions of copies. That amount of nanogram therefore gets replicated and is now ready to be sequenced, as we say it, in terms of deciphering the cancer biology in the case of cancer as such. So PCR reaction takes one gene at a time and amplifies it, and then we are able to quote unquote see in a molecular way the abnormality of that gene so that we can then target that gene. And unfortunately, cancer is a complex disease in which not one gene but multiple genes are acting. And PCR does not have the capability to act on multiple genes to amplify, which is why we go towards NGS or next gen sequencing, which takes care of amplifying not one gene, but multitude of genes from 50 to 100 to the whole genome. And we have about 20,000 genes in our genome, which then helps us decipher in a very sensitive way what's wrong with this cancer as such. We call it massively parallel sequencing to obtain millions, if not billions of DNA sequence reads at the same time. This gives us the advantage to take a small amount of the tumor biopsy and read these billions of DNA through very, very fantastic bioinformatic tools. We can do that for small number of genes by what we call a hotspot panel, which is what is called an amplicon or a PCR-based technology. The cost is very low for that. It requires very low amounts of inputs in terms of nanograms of DNA, and it's got a very fast turnaround. Or we can do a targeted panel of 100 to 500 genes by a technique called hybridization capture, where the cost may go up slightly, but the turnaround time is still quite quick. or we can do the whole genome of 20,000 genes where the cost is exorbitantly high. And we don't always need that kind of technology in a targeted panel when we know already which target genes we are looking for in that small amount of biopsy that we got from the patient. But all of this can lead to sometimes an error rate in which we may be able to have false positives or false negatives. And in technology has now come to the point where we can shrink that false error rate by doing some very fancy things by taking small amounts of the patient's cells, which are cancer cells mixed with normal cells, go through a preparation sequence by labeling them by what is called UMI, or unique molecular identifiers, or barcoding, so to speak, of a target gene sequence, and then reduce the error rate as such. And this is done bioinformatically by doing some preparations in a way that takes care of this whole sequence. Fairly complex way, but also has become fairly routine to do this now. This is no longer rocket science as such. When we sequence a patient's DNA, by doing this, we compare that with a reference genome, and in this case on chromosome 1, are able to point out if there is a mutation, if there is a copy number variation, or if there is a DNA rearrangement, and then are able to synthesize the knowledge that we need to treat this particular cancer. Back in 2000, when this process was started, to do this for the whole genome, it took $100 million as such. There's something called Moore's law, which says that over time, when technology becomes available, that cost will shrink because the technology has become available. But what happened in the case of biotechnology in the last 10 years after 2009 and 10 is that Moore's law was broken, and it did not follow, the cost decrease in genome sequencing did not follow this white line, but it precipitously decreased in cost to what we have for the whole genome in the US now. And if you do a targeted panel, that is much, much less in a matter of a few $100s in the US that it costs us now to take care of our patients by doing these kinds of sequencing as such. So the cost of sequencing depends on how deep you want to sequence to get to your particular gene of interest by doing certain assays like PCR based or targeted panel. And based on what clinical application you have a number of different techniques. This is very, very rapidly progressive and proliferative in the US. And in the last 5-7 years, it is now a matter of routine practice for us to do. It is amazing that this has happened so quickly, because this whole thing started by about 2007, 2008. And in the last 13, 14 years, we are here right now to take care of our patients. Now, that was solid biopsy. What about liquid biopsy? Liquid biopsy is a term by which we do the same thing that I've shown you in the last 5 minutes, taking a solid tumor biopsy, but in a blood sample. We believe, and in fact, we know now, that the solid tumor is constantly being shed into the blood of patients, and these circulating tumor molecules ascribable to the cancer can be purified and sequenced, so that the same information that we need to know from doing a solid tumor biopsy, we can get it in a blood sample now. And this has become even more proliferative in the last few years, when cell-free DNA-based nucleotides, or DNA and RNA, which are being shed into the blood by the cancer, can be easily caught and then studied for all those genetic defects I was talking about earlier. In fact, from 1966 to 2010 and 12, the number of publications in scientific media were pretty much stagnant. And you can see that by about 2013 or so, we have mushroomed the whole field of liquid biopsies that has taken over and become a dominant player to taking care of our patients as such. So liquid biopsy, as I said, sheds all these little molecules into the blood, and we can capture them in a blood tube of 10 to 20 ml of blood. And what we have seen actually is that even in non-diseased individuals, we can get what we call cell-free DNA in most normal individuals, as seen in this green bar over here. If you were to take a blood sample for a person without cancer, you can see that 0 to 99 nanograms of cell-free DNA will be found in these patients as such, or not patients, these normal individuals as such. But when you take it in colorectal cancer and pancreatic cancer, majority of the patients start increasing from 99 nanograms and less to 100 and more than 500 nanograms of cell-free DNA. Something is going on over here which the cancer is shedding. And it turns out that extra amount beyond the normal individual, which also secretes cell-free DNA, beyond that cell-free DNA, this extra amount of DNA that is being shed is because of the tumor. And this particular tumor DNA, we know now, has got between 170 and 180 nucleotide bases, or in other words, those letters ACTG, which is very qualitatively different from the fragments of 330 base pairs, which is the normal cell-free DNA that is shed into the blood. It is this 166 base pair fragments of the DNA in the blood that we capture and we sequence, and we catch the mutations of the cancer cell in the blood in the form of liquid biopsies. This has a very short half-life of 1 to 2 hours, or even some people say 15 to 30 minutes. And so we quickly have to capture it in our blood and proceed to the lab to sequence it quickly so that we can then translate our care for the patient. I'll give you some numbers over here. If in a cancer patient, I was to take 1 ml of plasma, which is a derivative of a whole blood sample, I will get about 10 nanograms of DNA in that 1 ml of plasma, approximately, average. That 10 nanograms of plasma, DNA, cell-free DNA, has about 3,000 copies of DNA as such. But those 3,000 copies are not from cancer. In fact, only 1% or more in advanced cancer, and in very early cancer, anywhere between 0.5 to 0.01% of these are belong to the cancer DNA of 160 to 170 base pairs. So let's assume we have 1% in this 3,000 copies, which are of DNA in the blood in 1 ml of plasma that we are looking at, which means we've got 30 DNA copies of the cancer cell in the blood that we have now got in front of us to study. And if we are looking at very, very small amounts of those 160 to 170 base pair fragments, like 0.01, we have less than 1 copy. So you need a very, very sensitive technology to get to these. One way of increasing the sensitivity of detection would be that we increase 1 ml of plasma to say 100 ml of plasma, which is impractical to bleed a patient for. But if you increase that, you can see how the numbers of the copies of which are ascribable to the cancer then can go up, even when it is a very, very rare amount of mutations that are present in the blood. So we need some very good molecular tools for that. And this process is the same, whether it is a solid or a liquid biopsy. We do molecular barcoding and sequencing, which then leads by bioinformatic analyses to a readout of the mutations that we are interested in and want to capture as such. So this is a non-invasive way to profile cancer, which is less invasive. It is low cost. It supplants the problem of tissue biopsies. It is very fast and it can capture the cancer heterogeneity. But the limiting and the factor and the biggest challenge is how much of the tumor DNA can we catch, which is also called tumor fraction. Depending on how much tumor fraction we can catch, or CT or circulating tumor DNA, we can apply the technology to catch and capture cancer mutations or copy number variations or other kinds of deletions in that blood sample and take care of our patients. So the applications can range of liquid biopsy from cancer detection to monitoring as well as treating those patients. Because it's easy to do a blood sample, we can do serial blood samples and apply liquid biopsies compared to a tissue biopsy as such. So now let me take the molecular tools and the tumor genome that I spoke about and give some precise application examples. Let me begin with an anecdotal case. This is a case of a patient, a true case from Memorial Sloan Kettering, of a 67-year-old woman who was diagnosed with breast cancer 3 years ago and treated appropriately, had a pathology, and all of a sudden, unfortunately, 3 years later, had 3 new lymph nodes that were metastatic and which appeared. They were biopsied and underwent immunohistochemistry, which is very standard as part of pathology to do, and it was found to be estrogen receptor positive. But because there was next-gen sequencing available for this patient, the biopsy material was sent for genomics. And what the biopsy material found was a bunch of genes listed over here, which were very, very mutated and altered in this biopsy specimen. It turns out, using bioinformatic algorithms, that these genes are classifiers of lung cancer much more likely than breast cancer. In fact, the tissue based on these results was sent back for more staining on immunohistochemistry, was positive for TTF1, which is classic for lung cancer, and it supports that diagnosis, and not for breast cancer, and was then gone on to treatment to receive treatment based on this genetic makeup. And that's where the power of NGS sequencing comes into play in our current practice as such. Another example of a gene called the androgen receptor in prostate cancer. In hormone-sensitive prostate cancer, which is treated with hormones, and in castrate-resistant prostate cancer, a gene called androgen receptor is very, very important. We know that. These little lollipops are basically mutations all over the gene, which can alter the biology and make the cancer progress. And we want to capture these. How do we capture this? We did an experiment and published it in which androgen receptor by PCR-based methods was diagnosed in patients in a liquid biopsy specimen, and was found to be that patients who harbored a copy number amplification or gain of this particular gene in the red curve over here did very, very poorly and responded poorly to treatment. But there were only 30% of patients out of the 100 plus patients who harbor these kinds of amplifications. The rest 70 did better than these ones, but they still died. That's because cancer is a complex disease and doesn't depend on one gene only. We need something which is NGS-based, so that beyond one particular PCR reaction, which is focused on androgen receptor, we can go to more genes at the same time in a cost-efficient way and find out the deleterious mutations and the alterations which we need to know to treat our patients appropriately. So we went and worked with a company, Predicin, who we worked with on our NGS panel to make an atlas of all the deleterious mutations that can be found in progressive states of metastatic prostate cancer, from an initial state called hormone-sensitive prostate cancer all the way to when it is treated with hormones and then recurs, called castrate-resistant state, and then is treated with more drugs, but unfortunately goes on to death. It takes about 5 to 7 years to travel from this point to this point. We were interested to know, using Predicin's NGS panel called Predicin Care, whether we will be able to find mutations that increase on this way and push this patient into death. So what we did was we created an atlas of alterations. We took 70 patients and followed them for 10 years when I was at Mayo Clinic, where I studied and led this particular study. Each of these columns that you see over here represents a patient undergoing NGS sequencing. Each of these letters over here are genes in that NGS panel. Each of these little dots, colored dots, represents either a missense mutation in that column or in that patient for a particular gene, and either a missense mutation or an amplification. And in untreated patients of hormone sensitive prostate cancer, going on to when they are treated with hormones, going on to when they're responding to hormones, going on to when they fail hormone treatments, and when they go on to becoming more progressive, you will see that on the right side of this landscape, suddenly the landscape becomes far more, unfortunately speaking, colored, which is to say there are more progressive alterations that can be picked up in the blood using this panel, which we can then study, like, for example, this particular gene called androgen receptor, and treat our patients depending on the presence or absence of that mutation. There are other genes in prostate cancer like P10, like RB1, and like TP53, which again, because there are multiple, we need NGS panels for. In fact, we published recently that those patients in blood who are subjected to this kind of NGS panel, when they are found to have detected to have tumor suppressor gene copy loss in RB1, in P10, and in TP53, and about 1/4 of the patients beyond androgen receptor amplification, which is bad again, then those patients are going to do very, very poorly and are not going to likely to have a response to drugs. This is by using an ultra-sensitive assay and by doing it in 10 ml of blood of the patient and deciphering these kinds of things. Now, we can tell about someone's survival by using these kinds of technologies. What about, can we treat them? In fact, we can treat them. We can detect these P10, PI3 kinase, AKT pathway aberrations, which are other genes which are altered in prostate cancer, in the blood of these patients. And based on that, we did a study which was from Australia and from Mayo Clinic, USA, in which we looked at hundreds of patients and were able to find these alterations in these genes, and found out that those patients who had these alterations did very poorly in terms of survival. But now suddenly there have been drugs developed for those patients who have AKT PI3 kinase mutations. And in one trial recently that was published in Europe, those patients who carry these mutations, when given that particular drug, seem to be benefiting from it. So again, we are matching pharma and drug pharma with alterations in these particular patients by doing a blood sample, by doing next-gen sequencing in the blood, and improving the patient's outcome as such. Here is another colleague of mine, Dr. Christoph Fanelli from Northwestern University, who did the same by detecting it in breast cancer patients by using two different assays, one from Predicin and one from Guardant, wherein these two were compared and found to have excellent performance for the Predicin assay in specific, if not for the Guardant assay also, and therefore again is now pushing the frontiers of trying to form an opinion of how to treat patients more smartly as such. Now that's good for the patient. Is it good for the cost to take care of these kinds of things? In fact, there's a study that has been published recently in which using NGS-based targeted sequencing of the tumor, whether in blood or in tumor, and detecting a positive alteration and giving all appropriate therapy actually was found to be associated with substantial cost saving, shorter time to test results, and more smarter treatment for the patient as such. It appears to be that precision oncology can have the power for a win-win situation for healthcare systems, as well as primarily for the patients and for the doctors and the nurses who take care of their patients as such. So the value of next-gen sequencing is that NGS and hotspot sequencing is here to stay. We have included them into our practice as such. They identify more alterations. We currently have no FDA-approved therapies other than those that are being developed or based on clinical trials, and they are becoming more and more prevalent as such. Upfront NGS testing seems to be the cheapest, broader molecular testing strategy, which we are using and in our practice as such. And it is a new era of genomic testing for cancer care, wherein multiple indications and adoption of these technologies beyond morphology and biopsies are changing the landscape of cancer care to the point of taking care of therapeutic matching, monitoring disease, prognostication of patients, of their survival, prediction of their response and their responses. And most of our national guidelines in the US and Europe now emphasize the use of molecular testing wherever it is appropriate and scientifically developed, and tests are available so that we can take care of our patients as such. I will stop on that brief run here and turn over to the forum for asking questions if there are questions for me.
Okay, thank you, Dr. Kohli. Very informative. Even for a person like me, I think the wealth of information provided is giving us a lot to look forward to. But a few questions just crops up.
Sure.
A question that was asked was this. You mentioned about 120 gene panel that you used for one of the research. Do all of them, when detected a mutation, have a therapy linked to them?
We did this back in 2016 and 17 when we were following our patients. We used that as our discovery set. What we do know now is that in that particular panel, we had used a panel of 120 genes called Precision Care, which I believe now has become 152 gene panel. But my point still is that based on that panel, we know that androgen receptor amplification in prostate cancer has now multiplied. Publications have told us that those patients who have that detected are going to not have good survival with our current armamentarium of treatments. It hasn't yet told us which drug to treat with. And for that, ongoing treatments like the one that I mentioned, PI3 kinase and AKT, so alteration, so that if you have a patient with AR amplification, and we know they're not going to do well using this panel, and in the same panel there are other genes like PI3 kinase and AKT, and there is a drug that targets that mutation or that alteration, in this case a deletion, then giving that particular drug to that patient becomes a feasible proposition for the future care of this patient. That's the kind of way science is unfolding more and more as we go forward. Many times we have to do this prospectively, and those prospective trials are ongoing right now.
Thank you very much. So it looks like there's positive movement towards drugs to treat, but of course the primary aim is you can't treat something you can't see, you can't detect. So the detection is more just as critical as the therapy. There's another question that's out here, because you mentioned about a comparison between NGS as well as spot testing, hotspot. Are there data to show the superiority of NGS versus hotspot towards detection and the success towards survivability?
So that's a very good question. The hotspot panel, which is about 50 genes or less, is amplicon based, and the NGS hybrid capture panel is typically more than that. The whole question's premise is based on what is your clinical indication and the reason to do whatever you're doing. If your reason to do is to do discovery and you don't know what you're looking for, because cancer is a complex disease and heterogeneous disease, you want more number of genes and a wider net to be cast in that situation. On the other hand, if you know already that there are a select number of genes that you want to target, then your clinical indication would be much more narrower, in which you have a smaller number of genes to look at, and then you would do a hotspot panel test. It all depends on what you want to achieve and want to do, and with the clinical indication that you have in mind.
Okay, just a follow-on question to that. What about the sensitivity of the biomarkers that are used? Are the sensitivity greater with an NGS panel as opposed to an hotspot, or that is not given?
The NGS panels are purposefully ultra sensitive. What I mentioned over there was that while they are ultra sensitive, you don't want to, because of that ultra sensitivity, call something, because that can impact downstream your treatment paradigm when you're treating someone with a purposeful mutation that you got on an NGS panel, and the person, it was a false positive. So in order to increase that sensitivity and the specificity, the UMI, or the unique molecular barcodes, that identifiers that have been invented, increase not just the sensitivity, but the specificity of your target panel mutation capture or alteration capture as such. And those technologies have now been very well woven into bioinformatic pipelines now, so that those our courage to accept those results depending on those pipelines is much greater every year in the last 5 years or so.
Thank you. Okay. I do have another question. Questions are coming in furiously. The other question is, we have spoken about late stage cancer, even your so-called, you shared on the lady with late stage cancer having not been detected. Now, is NGS just as valuable for early stage cancer?
Oh, that is a fantastic question. That is a fantastic question. And the reason why it's a fantastic question is, I showed you a table of the number of DNA copies and the sensitivity. I did that on purpose. And the reason was that when you're looking at early cancer, you have a very small volume of non-metastases. In fact, it's a very small cancer. It's not able to shed a lot of tumor DNA into the blood, so that the amount of DNA is less and less and less. The amount of DNA copies from the cancer is less and less and less. So you need something really, really, really specific and sensitive at the same time to get that. So there was recently, there have been publications in which screening for cancer has been implicated using NGS. And fortunately, they don't look at the DNA alteration. They look at something called methylation status. Methylation status of the DNA is basically an aberration that develops very early in the stage from normal to cancer, and is shed in small amounts in the blood as well as in the tumor as such. And these NGS technologies, by taking about 20 ml of blood and then using something called a technique called metabisulfite methylation assays, can detect the cancer early on. But there are two caveats to this answer I must implore upon you. Number one, we have been able to screen for cancer using these methylation biomarkers in liquid biopsies. We may be able to screen, but the word is not really screen. The word to be used over here is detect. And that word is a different meaning from screening. When you screen for cancer, you are basically trying to capture cancer early and treat it and change the course of its disease over time, over the lifetime of the patient. When you detect cancer, on the other hand, you're able to just detect, and you're not yet talking about treating the patient. So the trials that have been done so far, really, in my view, for detection of cancer. The screening of cancer to show tangible gain by doing this technology early on, and therefore having the gain in lifetime for the patient, is developing as science goes forward. Number 2 point, a few years back in Southeast Asia, there was a fantastic trial that was published in 2017 in the New England Journal, in which I think it was Singapore or I think it was Hong Kong, either one of those countries, which I can't remember, I'm sorry. But they were screening for blood biomarkers of nasopharyngeal carcinoma by looking at Epstein-Barr virus DNA in the blood of patients. Epstein-Barr virus is a virus that causes nasopharyngeal carcinoma. They did a population-based screening, and they found out that by screening for in the blood, by using sequencing technologies and capturing the Epstein-Barr virus DNA, not the tumor DNA, but the Epstein-Barr virus DNA, they were able to prevent the oncoming cancer in most of those cases. And that was a fantastic example of NGS technologies put in a population and a countrywide population to help stave off a national problem in Southeast Asia, where nasopharyngeal carcinoma is a big problem.
Yes, yes. Thank you very much. Just to follow on to that question itself, we detect at early stages, we still detect the mutation. And if there are mutations that have therapies available, so even though it's detection not towards treatment initially, because clinical studies are not leading towards so-called treatment at this point for early cancer, but those same mutations that exist can be treated using the available therapies that are out there, even at early stages. Am I right to say that?
That's a tough question to answer. And the reason is that if you have a standard of care treatment already in place, then you will opt for the standard of care treatment. Using these technologies is good, but you have to show that using these technologies will be better than the standard of care if we do that. And that's part of research. That is ongoing clinical trials that take place. But in the case that I showed you, which was in advanced cases, many times in cancers, we come to the end of our line of standard of care treatments and we shrug our shoulders. NGS sequencing is becoming very important and very helpful in those situations, in the advanced stages, when we can detect the mutations. And if there is a comparable drug to match with that mutation, we do end up treating. And sometimes, and most, or I don't know what percentage, but an appreciable amount of double-figure percentage of times, we help our patients. But there is a lot of research going on right now to answer these kinds of questions. And I'm going to leave it at that, because research is something that takes time to unfold.
Not to put you on a spot, but a question just came in. What they're saying is, currently the standard of care for stage 1 is more of a resection and removal of the cancer tumor. Now, if there are therapies that it's not resection based, even though they're not part of standard care.
Sorry, you're breaking up, Pradeep. There's a lot of static.
Okay, so the question would be, right now the standard of care for stage 1, stage 2, well, traditionally is to remove the tumor altogether. Okay, but the question they've raised is, if there are therapies that are more chemo-based, radiation-based, and based on early detection with using NGS, can they then opt not to go for the standard care and simply dive into the other therapies that is not invasive?
Well, that's a good question, but that will take a clinical trial to decipher that. But I will leave you with one other caveat on that question's answer. In 1970s, surgeons, by the way, the best tool we have to catch cancer early, and the second best tool we have is the knife, which to take care of early catching of cancer and to remove them. In the 1970s, in breast cancer patients at the National Institute of Health in the US and in Italy, two surgeons got together and said, we are catching patients early with breast cancer and we are removing their tumors, but they still come back over time, even though we have removed them completely. So let's add chemotherapy. What that did in the 1970s was that it improved the survival of those patients when the combination was with the knife and with chemotherapy. The same thing is happening now, where standard of care treatments, knife or other treatments, when combined with NGS panels or molecular diagnostics, when added together, yield better survival results. But this is a deliberate movement forward. It has to be done under the purview of research. It cannot be done willy-nilly as anecdotal cases. Otherwise, we will probably not do service to our patients.
Okay, we've room for time for one last question. NGS is gaining in popularity in US. How soon you think NGS testing could be widely used in India, and what is the gap?
I keep in touch with South Asia because I'm originally from there, and I give lectures and I talk to my colleagues and do lots of academic activity in India, and I've done some in other parts of Southeast Asia as well. My impression is that there are technologies, these technologies are becoming available. I'm not sure about the costness of those technologies, but these are available. In fact, we get patients from South Asia who come to us in the US already with NGS sequenced tumors. So these are becoming coming more and more into the play. I am not, like I said, sure about what the cost factor of these are in the local environment in South Asia, but the availability is there. You cannot duck NGS if you want to take care of cancer patients. That's the bottom line. This is a tool that will not disappear. This is a tool, a molecular profiling tool, which will be needed more and more and is going to be something that everyone needs to know about sooner or later if they don't know already. I'm sure many already know about it.
Thank you, Dr. Kohli. Now, there are a lot more questions. We will respond to those questions later on via email because we are running out of time. Dr. Kohli, very informative session once again, and thank you for the candid answering of practically every single question that we asked. I'm going to now hand over to Honorable Minister of Health of Mizoram, Dr. R. Laltangliada, who is going to share his thoughts as we close for the session. Minister, sir, are you there?
A very good morning for India and Bangladesh, and a very good evening for the, USA. Okay. So it's my, pleasure to participate this webinar, and I'm able to affirm the good works of Jakarta groups in bringing to this region a well-needed technology, at a very appropriate time today. Cancer has been accursed to the society, as we all know, and it is felt not just by the individuals, but the whole community and the nation at large. All of us are united in our fight against cancer, which has claimed countless lives and have taken a toll on the healthcare sector, as you all know. It is therefore a welcome blessing to have partners such as, the Jakarta groups who are going the full distance, in bringing to us innovative technology, such as the NGS-based liquid biopsy right at our doorstep. I'm really, really grateful to them. Needless to say, I'm confident that this informative session, stands as a testimony to the efforts of well-wishing partners in spreading their know-hows so that no one is left behind at this time. So as a Health Minister of Mizoram, I take one initiative. I just would like to bring to your notice that we are going to establish, a cancer super specialty hospital in Aizawl through the JICA funding, Japan International Funding Agency. Recently, we have settled with the government of India and with the JICA. And I can firmly vote at this time that with the growing number of cancer cases in my state, Mizoram, we stand united with companies like Jakarta Group in bringing to the ground this diagnostic that will serve our people. Thank you so much. Thank you.
Thank you, Minister, sir. Thank you so much. Once again, I'd like to thank everyone, and especially yourself and Dr. Kohli for making this session robust and very insightful. So till we meet again in the next session, I bid everybody farewell, and let's everyone come together and collaborate for a common unified good. Thank you, everyone.
Dr. I compliment you for what you are doing in Mizoram, and I hope you get lots of power to do that. This is a much needed thing.
Definitely, definitely. So I take this opportunity to wish again Zakaria the fullest, success, and offer the necessary support of the Health Ministry of Mizoram, okay, to jointly see the welfare and to take care of the people of Mizoram. So I take once again this opportunity to thank the speaker today for sharing valuable knowledge and your rich experience in the particular subjects. So with this, I wish everyone the most fruitful session. Thank you so much once again for the second time. Thank you.
Thank you. Okay, we'll see you again in the next session. Bye, everyone.
Bye.